Arskog-Scott syndrome, presented by Rachel Dowdoo. Arskog-Scott syndrome is defined as an extremely rare genetic disorder. This syndrome causes changes in the size and shape of certain bones and cartilage in the body, the face, fingers, and toes, and those are the features that are most often affected. This syndrome was discovered in 1970 by Arskog in Norway, and it was described as a syndrome associated with short stature with certain anomalies of the face, hands, feet, and genitalia in 7 males from one family. It was first described as a distinct disorder by Arskog in 1970 and further delineated by Scott in 1971. I'm sure you're familiar with this syndrome as you've seen some side effects or symptomologies such as the cleft palate. It's also known as facial dysogenitalia or dysplasia syndrome. Arskog-Scott syndrome is defined Arskog-Scott syndrome results from a mutation in the FGD1 gene, which is located on the short arm of the X chromosome. The inheritance pattern of this disease is typical of all sex-linked recessive mutations, those also found on the X chromosomes. Mothers who carry the mutated gene on one of their 2 chromosomes have a 50% chance of transmitting this disorder to each of their male children. Female children of female carriers also have a 50% chance of inheriting the gene and becoming carriers like their mothers. Although females rarely express the syndrome fully, female carriers may occasionally show minor manifestations. Affected males will also pass the mutation on to all of their daughters but none of their sons. Symptoms. The main symptoms of this syndrome are disproportionately short stature, abnormalities of the head and face, including a rounded face, wide-set eyes, slightly slanted eyes, drooping eyelids, a small nose, front-facing nostrils, an underdeveloped mid-portion of the face, a wide groove above the upper lip, a crease below the lower lip, folding of the top portion of the ear, or delayed teeth growth, and in some cases, the cleft palate or a cleft lip. Other symptoms may include a malformed scrotum, undescended testicles, small wide hands and feet, short fingers and toes, mild webbing of fingers and toes on a simian crease, which you'll really notice on this picture to the right. The first one on the left is the normal crease, and the one on the right is the kind of palmar crease, which is one elongated strip. Aarskog-Scott syndrome was first described as a distinct disorder by Aarskog in 1970 and further elaborated and delineated by Scott in 1971. This syndrome results from a mutation in the FGD gene, which is located on the sharp arm of the chromosome. It's extremely popular and common in males, but some features will be present in the female population. It's an extremely rare genetic disorder. Some additional features will be rounded face, a down slanted palpebral fissures, long upper lip with a linear dimple below their lower lip. Overall, what's most common genetically, Aarskog-Scott syndrome is a very interesting syndrome. It's extremely rare and no one knows the causes. They can see where it originates from on the biological and the molecular level, but as far as the just natural origins, no one actually knows. The genetics. The FGD1 gene specifies a guanine exchange factor, which is one of a class of proteins that act via an intracellular signaling network to turn on other genes at appropriate times during embryonic and fetal development. The specific genes and pathways known to be activated by the FGD1 protein are involved with the cell growth and differentiation of cells designated to become components of skin, bone, and cartilage. Additionally, the mouse has proven to be a useful animal model system for the study of this disorder, and the mouse data suggests that the FGD1 signaling specifically affects the development of several different skeletal cell types, including chronodoctides, osteoblasts, and mesenchymal prechondrocytes. Apologies. As stated earlier, there is no way to prevent this syndrome. All one can do is just become aware. Generally, for screening and diagnoses, the doctor will ask about symptoms and medical history and will perform a physical examination. The diagnosis of Aarskog-Scott syndrome is usually based on facial characteristics of children. The diagnosis can be confirmed by x-rays of the face and skull. Because this syndrome is caused by mutations in the FGD1 gene, genetic testing may also be used for diagnoses, generally in the in vitro stage. When the child is still inside the mother's belly, the doctor can assess through genetic testing whether the child is more likely to have this or if the child already does have this disorder. And then, unfortunately, what I've learned upon research is that parents can elect to evacuate the pregnancy prematurely. Empirically supported treatments and therapy. Unfortunately, there is no known cure for Aarskog-Scott syndrome. Treatment is limited to supportive treatment and surgical procedures to treat conditions caused by the disorder, such as inguinal hernias, cleft lip palates, or undescended testicles. So essentially, this is more of a presentation issue for parents or individuals dealing with this syndrome. In some cases, Orthodontic treatment may help certain facial and dental abnormalities caused by the disorder. Supportive treatment generally includes educational assistance for those affected with intellectual disabilities, including advice and supportive treatment for parents. Prevalence, prevention, and outcomes. There is no known way to prevent Arstock-Scott syndrome. Those with a family history of the disorder may be referred to a genetic counselor when deciding whether to have children. Additional risk factors. Mothers who carry the mutated gene on one of their two X chromosomes have a 50% chance of transmitting this disorder to each of their male children. Female children of female carriers actually have a 50% chance of inheriting the gene and becoming carriers just like their mothers. Arstock-Scott syndrome mainly affects males. Those at risk of inheriting this syndrome are male children of mothers who do not have the disorder but who carry the gene for it.